Hello, and thank you for joining us in our service learning project about the effect of psilocin on extracellular serotonin levels by David Lee, Chani Kim, and Teddy Kim. This video will demonstrate how psilocin, the psychedelic drug, affects the brain and its receptors. Understanding how this occurs may help further our knowledge on the effects and responses one has when taking psychedelic drugs, as well as receptor antagonistic drugs in general. First, some background. Psilocin is a hallucinogenic chemical derived from the Mexican mushroom Psilocybin mexicana. An antagonist is a biological molecule that opposes the effects of an agonist by binding to the same receptor because of its structural similarity. Here you see a sample diagram of what a generic agonist and sample antagonist relationship would look like. Serotonin is a neurotransmitter involved in producing melatonin to help regulate sleep, for balancing the appetite, for intervening with dopamine and noradrenaline, and for managing our sexual desire. Serotonin is a neurotransmitter that binds to a ligand-gated G-protein coupled receptor. This means that when serotonin binds to its target receptor, it acts as a ligand that changes the conformation of the receptor, which then phosphorylates a G protein to activate secondary enzymes or another enzymatic pathway. The G protein does this by attaching or coupling itself to another enzyme, which then activates or phosphorylates another enzyme, ultimately culminating in some sort of long-term cellular response. The many different types of 5-HT receptors activate different types of responses, ranging from effects on mood, behavior, appetite, and digestion. However, while there are many different subsets of the 5-HT G protein receptor family, they are all 7 transmembrane domains with an intracellular carboxy terminus and an extracellular amino terminus. Some important 5-HT receptors include the 5-HT1 receptor family, the 5-HT3 receptor, an anomaly of the other G proteins, and the 5-HT6 and 7 receptors. The 5-HT1 receptor family has receptors that inhibit adenylyl cyclase and encompasses the 5-HT1A, 1B, 1D, 1E, and 5-HT1F receptors. Here we see the 5-HT1A receptor. The inhibition of adenylyl cyclase prevents phosphorylation of other proteins. The 5-HT1A receptor is coupled to G proteins by 2 different systems. 1, the inhibition of adenylyl cyclase activity, and 2, the opening up of potassium channels, which results in hyperpolarization in neurons. In the hippocampus, 5-HT1 receptors are coupled to both effector systems and are thus vital for preventing unnecessary phosphorylation and in hyperpolarizing neurons. However, in the dorsal raphe nucleus, 5-HT1A receptors are coupled only to the opening of potassium channels, meaning that it is only located on presynaptic receptors and thus modulate the release of serotonin. Studies have shown that these receptors are in high concentrations in the basal ganglia. The 5-HT3 receptor is homomeric, meaning that its subunits are all identical. This unique receptor belongs to the ligand-gated ion channel family. This means that it does not involve activating a G protein pathway, but instead directly opens up an ion channel to allow in calcium or sodium. This causes the rapid depolarization of neurons. The 5-HT3 receptor, like other members of the ligand-gated ion channel superfamily, bears resemblance to another similar and important ligand receptor, the nicotinic acetylcholine receptor. Both of these receptors contain 5 subunits surrounding a central core, usually 2 alpha subunits, 1 beta, 1 gamma, and 1 alpha. These receptors are found in the peripheral ganglia and have been found to regulate depolarization of presynaptic transmission. The 5-HT4, 5-HT6, and 5-HT7 receptors are included in a family of serotonin receptors coupled to the stimulation of adenylyl cyclase, meaning that they may counteract the function of 5-HT1 receptors. These receptors have been located in the hippocampus. Psilocin is an analog of the 5-HT serotonin receptor, meaning that it has a structure similar to that of serotonin and combined to the 5-HT serotonin ligand site, acting as a structural homolog. Notice the structural similarity between psilocin and serotonin. They share a double ring structure with a pyrrole and phenyl group, as well as an ethylamine group. Serotonin has a primary amine, while psilocin has a tertiary amine. A study has found that psilocin specifically binds to the 5-HT1A and 5-HT2A receptors, meaning that it inhibits the effects of the 5-HT1 receptors to prevent adenylyl cyclase and phosphorylation. The significance of psilocin blocking the 5-H2A receptors is that 5-H2A receptor stimulation by psilocin is responsible for the psychedelic effects of psilocin. This is made apparent in the mechanism of the psilocin antagonist ketenserin, which blocks the 5-H2A receptor, meaning that psilocin cannot bind to the 5-H2A receptors. Thus, ketenserin acts as a sort of regulator of psychedelic activity produced by psilocin. Some effects reported by the use of psilocin include a mood lift, euphoria, sedation, close eye visuals and open eye visuals, hallucination, greater introspection, and greater creativity. Negative effects include fear, anxiety, confusion, dizziness, headache, GI discomfort, and nausea. This concludes our discussion on serotonin receptors and the potential effects of psilocin on them. As demonstrated earlier, the 5-HT serotonin receptors are mainly ligand-based G-protein coupled receptors, and psilocin may act as an antagonist to serotonin release through its structural similarity.